And thank you for listening to me at this late hour on a hot day. And if you go to the next slide, the normal disclaimer for you to read when at your leisure. Okay, so OBD, Oxford Biodynamics, is the world leader in what's called 3D genomics. We're currently based in Asia, in Malaysia, in Oxford, and in the States. We've been on the AIM market since 2016. The most important thing that you need to know about OBD is that we develop biomarkers, and that biomarkers are increasingly important in terms of pharmaceutical development for both pharma and biotech. And importantly, we are on 3 NIH biomarker steering committees in the States in oncology, immunology, and neurodegeneration. Over the years, we've worked in over 40 different partnerships showing how important this technology is. We have a very relatively strong balance sheet. As of the 31st of March, we have 13.9 million in cash on the balance sheet with no debt. Okay, so a biomarker is relatively simple. If you've got Alzheimer's, a biomarker might be a memory test. Our biomarkers are developed from blood. Effectively, anything that goes wrong with us, we can see systemically in blood. This is incredibly important because the failure rate on drug development is over 90%. And actually, 90% of all drugs do not work on 30 to 50% of people. The FDA, for obvious reasons, for cost reasons, and for the personalization of medicine reasons, are increasingly demanding that if you're going to have a drug, a registered drug on the market, you need to understand who is going to respond and who's not going to respond to that drug. And so that's effectively what we do with pharma and biotech. We give them the specific answer to the question about their drug in terms of response and non-response. This is critical not just to get authorization or registration, but it's also critical because you can have clinical trials where you can put responders and only those individuals who are responders into the clinical trial, and therefore increase your odds of success in that clinical trial. It also gives you considerable competitive advantage. You're able to show which cohort of patients your drug's most likely to work on. And that has not traditionally been the case. Pharma has traditionally sold to everyone, regardless of whether they understood whether that drug would work or not work. So there are different types of biomarker. There are obviously predictive. Does this drug work? There are what are called residual disease monitoring biomarkers. Can you monitor whether the disease has gone away? Sometimes diseases come back. There are prognostic biomarkers. In clinical trials, you really want to put people who have got a very aggressive form of the disease in your clinical trial, because you want to have the best bang for your buck in that clinical trial. And then the response biomarkers. These are we all respond differently to treatments. It's not true that we all respond within a week in the same way. We've worked in all of these areas for big pharma and for big biotech. And the insight that we had from Oxford is in the field of what's called chromatin conformation signatures. This is effectively DNA is arranged in a 3D structure around chromatin, which is called a chromatin conformation signature. And it's these CCSs that cause many of the interactions that take place within a cell. We all understand genomics or genetics. What we're really talking about is epigenetics here. Epigenetics is really the software that drives the hardware of the genetics. Another way of looking at it, if you were a poker player, the hand that you're dealt, the cards that you're dealt are your genes, but how you play those genes or your body plays those genes with the other players in the poker session is epigenetics. And it's increasingly clear that epigenetics is extremely important in terms of conditions and the result of those conditions that you may get. This slide is quite complex, but basically it shows that it's a paper that was written out of Cambridge, which basically shows that if you want to stratify, if you want to show red people against green people, you have to interrogate chromatin conformation signatures. And that is where all our IP and know-how is. There's a slide at the end in the appendix which shows our IP, but our IP is very broad-based and very important to the success of the company. It's not just us who are saying that chromatin conformations are important. There are over 50,000 publications, peer-reviewed publications, which indicate that chromatin conformation signatures are absolutely crucial in different therapeutic areas. In fact, even as long ago as 6 or 7 years ago, the paper on the right-hand side was written by McGill, showing that chromatin conformation signatures were ideal human disease biomarkers. The book on the left is produced by Harvard, where OBD has written the whole chapter on the key role of epigenetics in human disease prevention and mitigation. I won't go through all these indications, but the technology is very broad. When we spun out of Oxford University, we didn't know how powerful this technology was, this platform, which is called EpiSwitch. We've now worked in many, many areas. And on the right-hand side, where we're allowed to show who we're working with, you can see some of the logos from some of the big pharmas that we're working with or have worked with. Recently, we've even been asked by the Italian government to look at autism in blood. And if you'd asked us 2 years ago, we wouldn't have known that epigenetically you could see severe or non-severe autism in blood. And the process is relatively simple. We know where to look. One of the co-founders with myself came from the Sanger Institute. He was one of the first people in patent recognition. We know where to see these conformation signatures. That's in a way that no one else does. And then when we understand a plethora of alternatives in terms of potential biomarkers, we have a system that effectively produces a sensitive and specific and accurate test. We've all learned a lot recently about the accuracy of tests. So we believe that the world has gone our way with this COVID problem because people are starting to pay attention in a way that they weren't paying attention previously to diagnostic or prognostic tests. So specifically on COVID, we've recently announced that we are working with Glasgow University and the Scottish National Health Service. And basically the work we're doing is in two areas. We are looking at a particular drug and whether we can stratify patients that respond to that drug or don't respond to that drug. But more importantly, we're looking at cytokine storms. Can you tell from blood which patient will get a cytokine storm or not? And this is extremely important. And we have prior knowledge here because we've been working in the immunotherapy area. In fact, we've worked for the last 2 years in immunotherapy, and I'll briefly show you some slides later. So we understand that in certain conditions, one's immune system over-responds with catastrophic effects. And the other examples of where you might see this this immune response might be sepsis or in CAR-T treatments. So potentially by doing this work on COVID, which in itself is very important, the application of our technology will become even more recognized. Some examples of the work we do to give you a better feel for the importance of the technology. This is a contract we've signed with Mitsubishi in America. This is for a condition called ALS, which is a type of motor neuron disease. ALS is a terrible condition where traditionally you die within 2 years of diagnosis. If you look at the two graphs on the right-hand side, you can see the traditional way of measuring, the traditional biomarker for measuring whether someone has ALS is what's called an ALS-FRS score. You go to the doctor, he looks at your breathing, sees how you're walking across the room, other types of biomarker. On the other graph shows that OBD can tell the patient's condition from blood day one when they come into the GP's surgery. This is critically important, clearly, for patients who have a potentially two-year lifespan. But it's also very important to a company like Mitsubishi, which is trying to say to the FDA, this is actually a drug that's got authorization. We know which patients will respond to our drug and which patients will not. This trial is actually mandated by the FDA. It's called a phase 4. So they've already given authorization for this drug. And what they want to know is really who responds and who doesn't respond in terms of severity. Another example is work that we have just published last month, which is with Genentech and Roche. Roche owned Genentech. And the poster on the top left-hand corner was produced by Genentech 3 years ago, showing the validity of our technology. They then went and bought another platform for a multi-billion-dollar sum and came back to us recently because with this lymphoma, which is called DLBCL, it's extremely important to understand whether the patient is what's called GCB or ABC. Patients with ABC have a very poor prognosis, and as you can see, die relatively quickly. But there's also a third category, which those other technologies couldn't categorize, and it's called type 3. And at the bottom, you will see, this is Genentech and Roche's work, not ours. And the bottom, you can see that there's no difference between those graphs, or those lines on that graph. But with OBD's technology, EpiSwitch, you can see that we stratify this almost perfectly above. And so Roche and Genentech last month published this. The importance of this piece of work is clearly that it's Roche who are publishing it. And the difference between the three graphs at the top is that you can see under NanoString, which is a US company, and Fluidigm, there is a big black unclassified line. But with OBD, from blood, day one, there is no black line at all, no one unclassified. To give you another example, these anti-PD1 or PD-L1 drugs are what are called immunotherapy drugs. This is the great advance in terms of cancer treatment worldwide. There are over 5,000 trials in the States on immunotherapy drugs. If you look at the left-hand graph, this is what actually happened to the patients. So it was the clinical outcome. R standing for a response, and NR standing for non-response. We were given, on a retrospective basis, we were given these blood samples after the trial and were able to show to 82% accuracy from blood, day one, the likelihood of response or non-response. This is very important and was published by Pfizer, the Mayo, and EMD Serono just before Christmas. Next slide, please. And we really got into the public domain just last year. So all of this information that we're showing is relatively recent. And this slide is compelling. This was an article that was published in a well-known magazine called JAMA Oncology. If you want a perfect test, you want to make sure that you have no false positives and that you are 100% sensitive. So in other words, you can diagnose it 100% of the time, and you're not telling, for instance, a woman that she's got breast cancer when she hasn't got breast cancer. And if you look at this graph, this is shows some very large companies that and the results of their clinical trials in immunotherapy. OBD's balloon is one rather bigger than anyone else, which means that we've actually worked on more patient samples than anyone else, but also it is by far the most accurate in terms of sensitivity and how specific the test is. So this was a real public endorsement of the importance and robustness of the technology. So these are examples in the public domain of what biomarkers are worth to drug companies. If nearly all the drugs that we're working on are billion-dollar drugs, so they would expect to have sales of more than $1 billion a year. To put that in context, the largest arthritis drug from Abbott would have sales of something like $15 billion. And for that drug company to understand, in terms of stratification, what EpiSwitch is producing, the sums of money that they're paying here, which are in the public domain, are relatively insignificant against the sales. But this gives you a feel with some of the companies I've already mentioned, and obviously some of the companies that we've been working with, for the value of transactions that can be obtained by stratifying patients. If you go to the next slide, please. So in summary, we're the leaders in 3D genomics technology. We are the only commercially available 3D genomics biomarker discovery platform. We have a very robust and efficient technology and know-how. Most of our process is robotic and is based in Oxford. Our scalability is considerable. The robotic nature of what we do means that we've actually increased our capacity on the robotic side fourfold just in the last 2 years without putting on really very many people. And most importantly, there's a growing appreciation worldwide for the need to incorporate biomarkers in drug development. No longer can drugs continue to be sold without understanding response and non-response. And this has created a huge value for EpiSwitch. We have a strong balance sheet, we have unique and strong IP and know-how, and we have considerable momentum in a critical area of healthcare development, which has recently been demonstrated by Genentech, Roche, EMD, the Mayo, and Pfizer. Thank you very much.
Quite a lot of questions here around the same sort of theme, so I'm going to try and condense them into overarching, and it's about money, as you might guess from an investor audience, Christian, about, I guess, deal structure and how the technology is monetized, whether a client of yours, one of the big pharma or big biotech companies, are they paying per patient, paying per the drug license as a platform, and then who ends up with ownership? Right. Can you talk all around those subjects?
Yes, of course. So it's a license model. So what happens is that pharma, as an example, would ask us to stratify their patients between responders and non-responders. This might be, say, after phase 2, so they've seen efficacy, they've seen safety, they just don't know which patients are responding, which ones aren't. And they have to go to the next inflection point, which is a phase 3 trial, which is considerably more expensive. And what they will do is they will ask us to produce this biomarker panel from their drug in the particular condition that the drug is treating. And then we will come back with a panel of biomarkers, normally between 8 and 10 biomarkers, which are extremely accurate as to determine stratification. They will pay us for that work. And then as they will license the technology, and as the drug goes through its development phase, they pay us milestones as it reaches the drug's milestones. And then at the end of the period, they pay us a royalty on the drug. Now, this license model is actually very familiar to pharma and biotech. It's actually how they buy other pharma and biotech companies. So they'll pay you upfront money. Then as the drug develops, as sales develop, they pay you more money, and at different inflection points or different milestone points, they pay you further funds.
Okay. And you mentioned at the start you had 13.9 million in cash at the end of March,
Yes.
and no debt. So do you foresee, I guess, in the next, you can comment on this year, next year, the need to raise money, and then further to that,
So we don't.
when you might get cash positive, when you might get to that state?
So we're not allowed really on this forum to make projections, but if you look at the accounts, our net burn rate has never been really much more than £3.5 to £4 million. And we don't see that expanding dramatically over the next year or so.
Okay. I've had a question, and I'll read this out in its entirety, and it's relatively technical, so apologies to the questionnaire if I get anything incorrect, and hopefully, Christian, this makes sense to you. In the US, cancer patients are getting ctDNA next-generation sequencing tests for the genomic makeup of their cancer. Do you need to partner with one of these tests, and then in bracket, Guardian or Foundation? Can CCS work with these ctDNA tests? How do your epigenetic signatures differ from the epigenetic Guardian is introducing into their MRD tests?
Yeah, it's a very, very good question. So one of the earlier slides, the one from Cambridge, was showing that there are lots of other indirect modalities, sequencing being the biggest, huge industries which are out there in the marketplace. So these are all what you would call indirect competitors. There are no direct competitors to us. So the question is actually a very wise one, and it's a really very complicated. I try not to make the answer too complicated, but yes, it is possible to put our technology, our EpiSwitch, on these platforms. You've got to be very careful, though, that you're not giving them the solution that they need without making the economics. But it is true to say that that is a perfectly sensible potential route for us to expand the knowledge, certainly, of the value of this technology. It's a very good question.
Next one, quickie. Is it your long-term ambition to develop or to sell the company?
Oh, so there were three founders. One Soros took out a Russian. Soros took out of Russia before the wall came down, who was at Oxford. The other was an Indian who came under a Felix scholarship to Cambridge. And myself, our wish is for the technology to be globally utilized. This is so fundamental. The insight here. Clearly, the route for us, in the end of the day, your previous question asked about Foundation Medicine. That is an example of a platform company which was taken out by Roche for a very large sum of money, and is exploiting that technology properly. We would like this, the power of this platform to be utilized globally.
And I guess it's actually kind of following up to that. And another question: the technology sounds fantastic, but why is the market not appreciating this? Or can you point to anything why there isn't more demand for sharing?
It's a brilliant question. So the answer is I'm not sure that biotech in London is particularly well priced. And as the questionnaire has I'm sure knows, the market has been hit by a number of things, from Woodford to liquidity, especially AIM over the last couple of years. So there is an absolute lack of appreciation of value in terms of biotechs. Every company on the market that has got a COVID story seems to have gone up, but really robust platforms where the science is developed, where the financial risk for the investor, and I am personally an investor, is really just commercial at the moment. So AIM is not the best market to reflect value for these biotech stocks, unfortunately.
We've had a couple of presenters in the last month or so, biotech, healthcare companies who've talked about adding a US listing to kind of get peer valuation or to try and move into a more receptive market. Or would you see, is that a route you've looked at?
I'm sure that's an option. I mean, the best example I can remember is GW Pharma had a valuation of about £300 million in the UK for about 4 years, and I think is valued at 2 to 3 billion in the States now.
Yeah, it's a very good example. Christian, we're done. Thank you very much indeed for your presentation this evening, and thank you everybody at home for joining us.